Big data to understand big hearts. Single-cell transcriptomics provides insights into hypertrophic cardiomyopathy. A study published in Cell Reports. Hypertrophic cardiomyopathy is a heart disease caused by single mutations in the sarcomere, the protein structure that is responsible for contractions of the heart. The disease affects around 1 in 350 people. The defective contractile machinery can cause a stress response which leads to a swollen heart muscle. At the cellular level, cells can show disarray, increased cell size, and scar formation, also called fibrosis. This can lead to serious symptoms such as heart rhythm defects, severe heart failure, or sudden death. Patients with hypertrophic cardiomyopathy sometimes need myectomy surgery, during which a swollen part of the heart is removed that blocks blood outflow. In our study, we isolated and analyzed many individual cells, and determined the amount of expression from each gene in that cell. When we, for example, compared the expression of stress marker genes like NPPA and NPPB, ACTA1 and MYH7, we could confirm that cells from diseased hearts express more of these markers compared to healthy hearts. Single-cell data are displayed by UMAPs that display each muscle cell as a single dot. Distances between dots indicate similarity. As expected, a UMAP shows that hypertrophic cardiomyopathy cells, shown in red, are different from healthy control cells, here shown in gray. More importantly, when we look at the diseased muscle cells, we can further divide them into subpopulations. Differences between these subpopulations inform us about the molecular biology that underlies the disease. In this figure, we show marker genes for the identified cell clusters of the diseased heart. Cluster 3, for example, shows high levels of stress marker genes NPPA and NPPB, as indicated by the color coding of the UMAP. This subpopulation of cells might thus be extra sick compared to other cells from the HCM heart. To identify previously unknown genes that are involved in hypertrophic cardiomyopathy, we can look for genes with similar expression patterns as stress genes. Similarity in expression patterns can be quantified by correlation analysis. Indeed, genes correlated to stress gene NPPA, listed here in the rows of the right top plot, where squares indicate the correlation for a respective gene and patient, identifies multiple genes that are specific for the disease. This finding is validated by the bottom plot, which compares expression of several genes in HCM patients with healthy controls. This procedure can be repeated for multiple genes of interest. Here, plots are shown for XIRP2, a cluster 2 gene. Look at the plot on the right. The left 5 columns show strong correlations in patients, whereas the right columns show weak correlations in healthy control hearts. This indicates that these correlations between genes are disease-specific. Generally, gene-gene correlations are very interesting, as genes with similar expression patterns over the subpopulations are likely to be regulated by similar factors. Moreover, a similar expression pattern suggests there might be functional links between these groups. Thus, correlations tell us something about the molecular mechanisms underlying the disease. Here, the correlations between virtually all genes for 3 patients are shown in a heat map. Clearly, there are groups of genes that show more correlated expression within a group of genes, compared to genes outside the group. These could be functional modules related to disease processes. Moreover, these groups can be used to identify regulatory factors that drive the disease, by looking for regulatory factors that interact preferentially with genes in these groups. Indeed, some identified regulatory factors we observed were also identified through independent experiments that were published earlier. Importantly, we also identified previously unknown regulatory factors potentially related to hypertrophic cardiomyopathy. In addition, we assessed gene expressions that are correlated to cell size. You can read all those outcomes in our publication in Cell Reports. We have quantified gene expression in single muscle cells of diseased and healthy hearts. By looking at subpopulations, correlations between gene expression, analysis of transcription factors, and analyses of cell size, we can identify genes and regulatory factors that will help us to understand molecular mechanisms behind heart disease and develop new medicines to save lives.